Hi everyone, and welcome to today's lecture on microbial growth for the part 1 video, and then in the part 2, the second video, you'll encounter control of microbial growth as well as a few slides covering some diversity of metabolic pathways and functional diversity. Now, when we talk about microbial growth, the first thing you're going to want to think about is what exactly is required for microbes to grow. When we talk about requirements for their growth, there are two main categories, okay? There are the physical requirements, things such as temperature, pH, how salty the environment is. And then there are the chemical requirements, and these include things like CHO, as I like to call it, carbon, hydrogen, and oxygen, things like nitrogen, sulfur, phosphorus, and our various minerals as well. And when we talk about the chemical requirements, in a little while we're going to go one by one and basically see what each of these is needed for in the cell. But before we get to that, we're going to go through the physical requirements, starting off with. Now, temperature is going to be the first requirement that we talk about. And when we talk about temperatures, there's a term I want you to know, which is cardinal temperatures. Every organism, including you, have cardinal temperatures. And what that means is you have a minimum, a maximum, and an optimum growth temperature. If you are at your optimum, great. Your cells are happy, your proteins, all of your biomolecules, everything's happy. When you end up outside of that optimal temperature and you go beyond your minimum, beyond your maximum, then you start to encounter various problems. And we're going to talk about these in the next slide a little bit more as well. But basically, when you are below minimum, your enzymes, which are a type of protein, a lot of your metabolic reactions, they slow down and they will almost, in some cases, stop. When you are above your maximum cardinal temperature, you end up having issues with your biomolecules, particularly enzymes, that end up denaturing at high temperatures, meaning they unfold, they don't work properly. That's very problematic, and cells start to die, and ultimately the organism can die. So we're going to go more through the slide. You'll keep hearing the terms bacteriostatic, which is really cold, usually in colder, below minimum temperatures, where everything starts to slow down or stop, hence the term static at the end, or stasis, if you think about it that way. Whereas above maximum, really high temperatures, like for instance cooking things on a flame, that we're going to keep mentioning as bactericidal, which means you're killing the microbe. Cells are dying, you're killing. Okay, again, these are terms that are going to keep coming up throughout this lecture, as well as lab and some other lectures too. So we're going to start with temperature. And when it comes to temperature, there's a term I want you to remember, which is the cardinal temperatures. Okay, every organism, including us, has cardinal temperatures, which means every organism has a minimum, a maximum, and an optimal temperature. Okay, so when you hear cardinal temperatures, think the minimum, maximum, and optimal temperature for that organism. Now, if there is a temperature that is below that organism's minimum temperature, then that organism is going to be considered metabolically inactive, okay? And the term you'll hear associated with that you have to remember is bacteriostatic. We're going to see this in some of the labs that we do and also in other lectures. So whenever you hear bacteriostatic, think stasis, staying still. Okay, slowing, stopping. There is no more growth of that organism. Okay, now when you think of that, think of what happens if you end up below temperature. Or if you put food below temperature, think of things like freezing, okay? So very cold temperature stops growth. Whereas if a temperature is above the maximum, what does that mean? Well, very hot temperatures, they're going to destroy. They're going to cause death, okay? That's why heating things, okay, cooking food is done, because that kills microbes, okay? And what we call that is bactericidal. So when you hear "cidal" at the end of a word, like genocidal, suicidal, homicidal, that means death or killing, okay? So high temperatures kill, cold temperatures stop growth. But notice, what am I not saying about cold temperatures? They're not killing. Okay, so always remember that when you put food in the fridge or the freezer. You don't want to keep it there forever because you are not killing the microbes. You're just slowing down their growth, but they're still there and they can still eventually grow. Okay, so now when we talk about temperature requirements, we have three main categories. You have to remember these terms. Okay, we have psychrophiles, we have mesophiles, and we have thermophiles. Okay. The psychrophiles, I like to call them the psychophiles. They like really cold temperatures. Okay, you can see on the slide that I put the range from -5 to 20 degrees Celsius. Now, because they thrive in cold temperatures, you have to ask yourself, where might I find a psychrophile? Well, growing in the refrigerator, right? So it makes sense that these guys will cause food spoilage in your fridge. Okay. The next category, I want you to put stars next to it, circle it, highlight it. That's mesophiles. When we talk about mesophiles, they like moderate temperatures, and I have the temperature range 20 to 45 written there. I want you to put little stars next to that temperature range and ask yourself, what falls in that range? Why is that range so important? Okay? Ask yourself, well, what's 20 degrees Celsius? Think about it. That's room temperature, isn't it? So these guys can grow at room temperature. What else is in that range? Well, 37 degrees Celsius. Where have you heard 37 degrees Celsius before? That's body temperature, right? So not only can these guys thrive at room temperature, but they can also thrive at body temperature, which means what are mesophiles going to be causing? Human illness. Okay, so it's very important for you to remember the significance of that temperature range for mesophiles. The last category is thermophiles, and when you hear thermo, you think heat. So these guys like high temperatures, greater than 45 degrees Celsius. And anytime you categorize a thermophile, I do not want you to use just the word thermophile. You have to be specific. You have to say, is it facultative? Is it obligate? Or is it extreme? Now, the easiest one to define is think about obligate first. Ask yourself, if you are obligated to do something, what does that mean? You're required to do it, right? So obligate thermophiles require high temperatures. They will only grow above 45 degrees Celsius. Okay? Whereas facultative, they are not obligated. They're not required to. Okay? They just like high temperatures, but they can also grow below that. Okay? So a facultative thermophile will grow above 45 degrees Celsius, but also be able to grow below that, which means these guys can also grow at 37 degrees Celsius, which you have to remember, that's body temperature. Okay, so they can cause a problem for us, the facultative thermophiles. Then the last one is extreme thermophiles. And if anything's extreme, that means crazy high. Okay, so extreme thermophiles you will find above 80 degrees Celsius. Okay, now to help you visualize these, we have a figure on the next slide. Okay, so when you look at this figure, you can see in order we have the psychrophiles, the mesophiles, and the thermophiles. Now you also see a curve for hyperthermophiles. What do you think those are? Well, those are the extreme thermophiles we mentioned, okay? So when you look at this, it's just to help you better visualize, but you have to understand what's on the previous slide more importantly, okay? Now, in addition to temperature, you also have pH as a critical physical requirement. Now, most organisms, most cells, just like even in our body, they prefer a pH of 7. Okay? What is a pH of 7? That's neutral, right? Now, as you can see by the figure, the little picture or cartoon that I put on this slide, the reason why cells prefer a neutral pH is that if you have the wrong pH, well, enzymes do not like that. And if enzymes get denatured, then a cell will not be able to function. Okay? Now, some cells, some microbes, can survive out of that neutral range. If they like acidic pH, okay, if they can survive at very low pH, then these guys you would call acidophiles. And you have to ask yourself, where might you find some? Okay? Think about your body. Where are the acidic regions of your body? The stomach, right? That's a big one. Okay, the GI tract, your gut. So any of the organisms that you might have thriving there, they would have to be acidophiles. Now, there are quite a few acidophiles that we may encounter, but on the opposite side of that spectrum, alkaline environments, there tend not to be many microbes that can survive alkaline, and alkaline is just a fancy way of saying basic, right? So when you think of it that way, that's why a lot of disinfectants are alkaline, okay? Things like ammonia, because even though we have a lot of microbes that can survive acidic, don't really have many that can survive alkaline, okay? Now, in addition to temperature and pH, we have osmotic pressure. And yes, that little GIF cartoon in the corner, that is one of my favorites. Okay. Now, when we talk about osmotic pressure, you have to think about salt. And this can be a reminder of when we drew out or thought about hypertonic and hypotonic environments. Okay. If there's tons of salt in an environment, what happens to that cell? Well, salty, you dry out, right? Shriveled up. Think of eating something really salty. Even though it's delicious, you need a lot of water after. Okay? And cells do not like that. It can be very dangerous for them. So when we're talking about salt content in the environment, if a bacteria or if a microbe is capable of surviving in high salt, we call them a halophile. Now there's a magic number I want you to put a star next to and write down, and that is 3%. 3% is the salt content value that I want you to remember, okay? If a microbe can grow above 3%, that's when we will categorize it as a halophile, right? If it's below 3% growing and it cannot survive above 3%, if it dies above that, then we consider it not to be a halophile. That's just normal bacteria, okay? The normal range for bacteria is 0.5 to 3% salt. So above that is when it becomes a halophile category. Now, just like we categorize the thermophiles, you also can categorize halophiles based on their salt preferences. Okay, it's the same terminology. You have obligate, you have facultative, and you have extreme. So ask yourself, what did we say about obligate? Well, it's obligated. Okay, so obligate halophiles are obligated or required to have high salt. Okay, you will only see them growing above 3%. They will not grow below that. Facultative, on the other hand, they can grow above or below 3%. Okay, so if you see something growing above 3%, that tells you it's a halophile. And if it's still growing below that, it is facultative. If it is only growing above, it's obligate. And then the last one is extreme, which means a concentration of about 30%, even 20% sometimes we consider extreme halophiles. Okay, so you can visualize this a little bit better when we do the laboratory exercise on it. Now we're going to go through the chemical requirements, okay? Now with each of these, you have to ask yourself, what would a cell need this chemical for? So the first category of chemical requirements, CHO, which is carbon, hydrogen, and oxygen, these are critical. Put a little star next to the title of this one because these three are necessary for all cells to build their biomolecules. And when we talked about biomolecules, that was nucleic acids, carbohydrates, lipids, and proteins. Okay? Without carbon, hydrogen, and oxygen, these cannot be made. Okay? Then you also have nitrogen. Nitrogen I write out as NaN, N-A-N, to remind myself that nitrogen is required for amino acids and nucleic acids. Okay? So when you see those words, you have to ask yourself, well, what is this cell actually building? Well, amino acids, that's proteins, right? That's the building blocks of proteins. And nucleic acids are for DNA, RNA, and ATP. Okay? So nitrogen is needed to make proteins and to make things like DNA and RNA. Okay, so very important, nitrogen. Then we have sulfur. And with sulfur, I want you to circle methionine. Circle the word methionine or met. This you should remember from when we did the genetics lesson. Okay, F-met was the initiator amino acid, which means methionine is the first amino acid of any protein. Okay, so without sulfur, which is part of methionine, cells would not be able to build any proteins. Okay, so sulfur is very critical. Then you have phosphorus, and phosphorus is important for nucleic acids, phospholipid bilayer, and ATP. Okay, so again, whenever you see nucleic acids, you have to tell yourself DNA, RNA, and ATP. Okay, and then the phospholipid bilayer, that's the outer membrane of a cell. That's what gives it its structure and protects it. So without phosphorus, you would not have a cell. The last of these chemical requirements are potassium, magnesium, calcium, and iron. Okay, so we have some minerals here. These are all cofactors. Okay, so you should remember that from our previous lectures. And when you see cofactors, you can underline factor and remind yourself the little trick of factories. Think of a factory with metals and minerals, okay? So these are inorganics that help activate enzymes. Enzymes would not function without them. Okay, what was the other word that we learned with this? Cofactors are inorganic, and coenzymes, which are vitamins, are organic. Okay, both are needed to help enzymes function. The next thing to mention is oxygen. So this is different from when we said carbon, hydrogen, oxygen. Okay, when we talked about those, we're talking about, for instance, breaking down foods, plants, to get that piece of oxygen. The oxygen on this slide, however, is talking about atmospheric oxygen. Okay, taking atmospheric oxygen. Big difference, okay? So they all need the oxygen of carbon, hydrogen, oxygen, but they do not all require atmospheric oxygen. And in fact, some bacteria get killed by atmospheric oxygen. Okay, so I write out a few different categories here for explaining the oxygen requirements of bacteria. Okay, so the thing about oxygen or atmospheric oxygen is that it causes free radicals. And free radicals in the atmosphere or in an organism's cells are unstable. Okay, free radicals, I'm sure you've heard this as a bad thing because they are unstable and they steal electrons from other molecules. Now, if they're stealing electrons from other molecules, what happens to those molecules that lost their electrons? Well, now they're unstable. Now they want to steal more electrons. Okay, so you have this chain of events of atoms becoming unstable, which is a big problem for a cell, and that can kill it. Okay, now if a cell has what's called superoxide dismutase, okay, SOD, I have it written in blue on the slide. You can circle and put stars next to that word, superoxide dismutase. Okay, if a cell can make that enzyme, then it can break down or convert free radicals to safe oxygen. Okay? A lot of times it also uses catalase and peroxidase. Now you don't have to memorize or know the chemical formulas or the biochemistry behind this. Simply know that the enzyme superoxide dismutase, or SOD, converts dangerous or toxic free radicals to safe oxygen. So if I'm a cell and I can produce that enzyme, then I can make the atmosphere safe for me. Okay? Any of the ones that can do that can survive atmospheric oxygen, and they are called aerobes. Okay? Aerobes, either obligate or facultative. If, however, a bacteria cannot produce that, does not have that, it is called an obligate anaerobe, meaning it requires a lack of oxygen. If it encounters oxygen in the atmosphere, what will happen? It'll die. It will die because the free radicals will make it unstable, and it has no way to counteract the free radicals. Okay, so aerobes produce superoxide dismutase and protect themselves. Obligate anaerobes do not. You also see a couple of extra categories, which are aerotolerant or microaerophiles. These guys can survive in oxygen, meaning that they do have the enzymes such as catalase to protect themselves, but they don't actually use oxygen, or they need it at a low concentration. Okay, but what I really want you to remember is aerobe versus obligate anaerobe, the concept of superoxide dismutase. Okay. Now, beyond the requirements for growth, you have to think about the ways in which bacteria grow or microbes grow. And one way that they grow, some of them, is as biofilms. And when you hear the word biofilm, there are two things I want you to remember. One is the definition, which is that a biofilm is a whole bunch of different species of bacteria that stick together into this big collection, working together, protecting each other, all different species, okay? Which means that they are a lot harder for us to destroy. An example of that, which is the other thing I want you to remember about biofilms, is plaque on the teeth. Okay, plaque on the teeth. And a lot of these guys in biofilms, what they will have is a glycocalyx, which we already mentioned in previous lessons. That is basically that sticky slime layer around them, a capsule around them, which is why they're able to stick to things such as your teeth. Okay, but for biofilms, remember it's a whole bunch of different bacteria working together, and the example is plaque. Now, when we talk about the growth of bacteria, you also have to think about how they replicate. Okay? The way that bacteria replicate is through binary fission. So as you can see in this figure, one cell splitting into two. Now notice, what do they have to do before they split, before they undergo that mitosis? They have to replicate their DNA. Okay, so we mentioned that when we talked about the genetics lesson. Okay. Now, the next slide is very important. The 4 phases to bacterial population growth. I want you to put stars next to this slide You should be able to understand the 4 phases. You should be able to name them and to make a key point about each one, okay? So the 4 phases are the lag phase, the log phase, stationary phase, and the death phase. And the way that you can visualize them is on the next slide, and I'll put the next slide on the screen, but I will explain what we have on this slide as we're looking at the next slide, okay? So looking at our 4 stages, like I said, you have to know your 4 stages: lag, log, stationary, and death. Okay. Sometimes, as you can see in this figure, we call the exponential, the log phase, we can call exponential. And sometimes they'll throw in at the very end a long-term stationary phase, but you don't have to know that. You have to know lag, log, stationary, and death. So what happens at the lag phase, you'll notice at that phase you don't have many bacteria yet. Okay? You do not have any division occurring. And instead, the bacteria at this point, they're just getting used to things, okay? They're getting their enzymes ready. They have to start transcription, translation. Remember, we said these things take time, okay? Once they finish with that, then they move on to the log phase. And now is when you have binary fission, okay? Since you have binary fission, the numbers are increasing exponentially. Okay? But this is only when conditions are favorable. Now, circle on your slides the fact that log phase, the bacteria are metabolically active. Okay, circle or write metabolically active in the log phase. That's very important because if a bacteria is metabolically active, that is when it is sensitive to drugs and antibiotics, okay, to your treatment as nurses or as doctors, okay? Once it's made it through there, you'll notice that our curve starts to flatline up at the top, okay? That's now the stationary phase. The reason why it's flatlining is because the cell death is equal to the cell division rate, okay? And you have to ask yourself, why is this happening? Why are they starting to die at the same rate that they're dividing? Well, look at the figure. You notice space gets crowded, okay? When there are too many bacteria, they can't properly survive. And when there are too many bacteria now, they release a lot of waste. Think about it. They're not just eating and growing and replicating, but they're also excreting, which means they now have a lot of waste and gas and toxins built up there. Okay, so they start to die off. And then as they start to die off more than they replicate, you notice you get the plummeting curve, which is the death phase. Okay, so now the cell death rate is greater than cell division. And that's because they've used up their nutrients. Okay, they've used up the nutrients. There are too many wastes built up and too many bacteria there. Okay, so you have to know lag, log, stationary, death, and what's happening at each one. Okay, so lag is the prep phase. Log is when they're metabolically active and replicating. Stationary is when you have too many of them building up. And death phase is once they're now used up nutrients, there's too many wastes, and they're dying off. Now, when we talk about growth requirements, you have to ask yourself in terms of our labs, in terms of medical research, well, if we know the growth requirements, then how can we culture these bacteria? And that's where culture medium comes in, which you've worked with in the lab already. Okay? If it is in the plate form, the solid form that we've worked with, which is kind of like a jello form, that is called agar. Okay? It's got solidifying agents. The media also needs to have extracts, and these are important for giving it all of those chemical requirements we mentioned earlier. So The yeast, the meat, the plants, they give it things like the carbon, sulfur, the phosphates that we talked about earlier. And ultimately, that is why the media is so important. It's giving the bacteria what they need to grow. Now, media is also very valuable to us in the medical field and the research field because we have different types of media. And these different types of media allow us to do things like diagnose. The two main categories that really help us in terms of diagnosing or working with various bacteria are selective and differential media. And you have to know the difference between selective aspects of media and differential aspects of media. Okay, so put a star on this slide because you will be asked about it again in the future. So first we have the idea of selective media. Selective means you're selecting for something, okay? So you will either see growth or no growth. There will be inhibitors in this kind of media that block certain bacteria from growing, okay? So the picture that we have on the slide, this plate here, that's an MSA plate, mannitol salt agar, okay? The fact that it has salt in it is what makes it selective, okay? So if I ever ask you the selective aspect of an MSA plate, well, that's the salt, okay? Because as you know, only halophiles can grow in high salt. Then what else do you notice about that plate? Well, you notice you see different colors, okay? So when you see the word differential media, I want you to underline different in it. Okay, underline different in it and write for yourself different colors. Okay, so the differential aspect of the plate that you see on that screen is that red media turns to yellow if the bacteria can ferment mannitol sugar. Okay, and so what you notice is on an MSA plate, Staph aureus turns the plate yellow. And that's critical when you want to distinguish pathogens, because when you look at that plate on the screen, well, Staph epidermidis, it looks red, but Staph aureus looks yellow. Well, what do you know about Staph epidermidis? Epidermis, skin. It's a normal flora on your skin. Whereas Staph aureus, well, when you hear MRSA or MRSA, you get scared because Staph aureus is a bad pathogen. Okay, so you want to have media that can quickly tell you which of these two you have that your patient is dealing with. Okay, now a lot of plates are both selective and differential at the same time. So you'll notice in the picture, MSA plates are selective for salt at the same time that they change color for a differential aspect. Okay, but know the difference between selective aspect and the differential aspect. Another type of media that I want you to be familiar with is reducing media. Now, reducing media, you don't have to memorize or know the actual mechanism that it's working by. I just want you to be aware that reducing media exists, and specifically, sometimes you'll hear the term thioglycolate. Okay, that's in the second bullet point here. Whenever you hear thioglycolate or reducing media, simply know that that's for growing or categorizing anaerobic bacteria. Okay, bacteria that cannot survive in the presence of oxygen. Okay? And what's cool about the thioglycolate is because it removes or uses up the oxygen in the media, we can grow or put bacteria into a tube of this, and we can see, does the bacteria stay at the very top and grow, meaning that it wants the oxygen from the atmosphere, or does it only grow at the bottom where the thioglycolate has used up oxygen? Okay, so very valuable to have reducing media. But just as soon as you hear reducing or thioglycolate, think anaerobic. The next thing that I want you to know is how to preserve bacterial cultures. So again, we're not going to go into the crazy mechanisms of these, but I just want you to be aware that sometimes we need to preserve bacteria so that, for instance, we can ship it to labs, or so that you can send out your patient's culture to other research facilities to try and figure out more about what is ailing them. And so two of the ways to preserve bacterial cultures are deep freezing or lyophilization. And lyophilization is just a fancy way of saying freeze and dry out. Now, what I want you to know for here is, just like we mentioned in the previous slide, I want you to be able to know that these terms exist and what they're for. So if you hear lyophilization, or if you see that word, instantly think preserving, okay? Okay, so that's it for the first part of the lecture, focusing on microbial growth. Now in the second video, we're going to focus on controlling that growth. A lot of the things we encounter in that second part will be very relevant now that you have lived through a time of pandemic. And then at the end, we will also discuss a little bit about metabolic diversity as well as functional diversity. So you can take a break, or you can head on over to that second video now.